Den neste foredragsholderen som skal presentere her i dag, det er Andy Taylor, som er seniorforsker ved James Hutton Institute. Andy har i lang tid jobbet med hvordan sopp oppfører seg i miljøet, spesielt hvordan sopp er essensielt for mange planter. Foredraget hans er på engelsk, og titelen er Fungal Assisted Regeneration of Atlantic Woodlands. Fritt oversatt så kan man si at det er soppassistert regenerering av atlantiske skoger.
So thank you very much for the invitation to talk about some of the work that we've been doing. And it's comparable to some of the systems that you have on the west coast of Norway, so it may ring some bells with people. And what I'm going to be talking about is fungal-assisted regeneration of some of our Atlantic woodlands. So I just wanted to point out a few things, first of all. Not all of Scotland is a treeless, barren land. Neither is it one big citrus spruce plantation, although sometimes it feels like that. So we do, in fact, have some existing ancient woodland. Some of it is Caledonian pine forests, and there are some fragments of this left. There's about 86 fragments, some of which are only, unfortunately, a few trees, others are several thousand hectares. But what I'm going to talk about today is really forests that we share with Norway, and that's temperate rainforest. And if I just blow up the top part of the map here, I've circled Scotland, showing the green areas on the west coast, and you can see also the green areas on the west coast of Norway. So they're certainly comparable systems. And some of them you will be familiar with. There's hazel woodlands, mainly the tree species Corylus avellana, and it's famous for this fungus on the right-hand side here, which is, in English, it's called hazel gloves, Hypocreopsis rhododendri. And it's one of the flagship species which has been used to conserve the hazel woodlands on the west coast. The other, perhaps more famous woodland on the west coast is the oak woodlands, and these are really ancient woodlands that have survived for various reasons. And they're Quercus species, birch, betula, and also quite often Fraxinus excelsior, the ash tree. And these are incredibly rich in lichens and bryophytes. And as yet, we know very little about the fungi, the below-ground fungi in these systems, but it's something we are working on. The system I want to focus on today is really the Atlantic pine and birch woodlands. And the pine is Pinus sylvestris, and then the birch is the Betula species. And these are very rich in fungi, particularly mycorrhizal fungi, and it's really these these associations that I want to talk about today. So the site which I'll be talking about is this mountain here. By your standards, it's not a mountain. It's only 530 meters high, but it's Ben Shieldaig, away over on the west coast. And the total area is about 4,000 hectares. And it was bought by a charity called the Woodland Trust in 2019. And the aim of the trust is to reforest the mountain with woodland that would have been there before it was cleared. And we have a project with them to develop ways of working with nature to promote the regeneration of woodland on the mountain. Now, there are two existing areas. On the left-hand side there, you've got Atlantic birchwood, and on the right-hand side, it's a remnant of Atlantic pine forest, and it's the most westerly pine forest, old pine forest that we have. Now, this all looks very nice, and this is in fact the view which the Woodland Trust used to raise support for the project, but the reality of the situation on the ground is really rather different. And this is where we've become involved. Because if you look from the other direction, this is looking north. The nice picture earlier was looking south on a very nice day. This is a much more typical day, very misty, and the ground here is very open. It's been heavily grazed and repeatedly burnt over the years, and it's very open and really quite in poor condition. And this is what they're hoping to recreate the woodland on. So the pine wood that you saw in the last picture is the southern end of it. So it goes along the face a little bit, but not very far. Most of the ground is 90% wet or dry heather moorland, very coarse vegetation, and also intermixed with fairly poor grassland, including Millennia that we'll see in a minute. Okay, so it's very different. It's poor ground that's been grazed and burned. So how do we go from this bare ground to woodlands? Because it's quite a challenge. There are very few established trees. You can see one up at the top there on the crag, but there are very few mature trees along the slope to supply seeds. So how do we go from bare ground to something that looks like this? This is the ultimate aim, is to get something regenerating, which is a mix of birch and pine. So what we started doing was looking at the mycorrhizal associations across the site. And what we've done is to generate maps, effectively, of the mycorrhizal associations on the vegetation across this site. And I really wanted to give you a reminder why mycorrhizal associations are important. You probably know, but I just wanted to say a few things about them. So most of the trees which are used for regeneration or for outplanting are in fact devoid of mycorrhizal fungi. They go out from the nursery and they have no mycorrhizal beneficial fungi associated with them. And they have to acquire them after they have been planted out. And if the seedlings are going into a system that has lots of mycorrhizal fungi, suitable ectomycorrhizal fungi, then it's not a problem. But in order for these fungi to really help the seedlings survive and to become established, they really need to be there. And that's one of the issues that we face at Ben Shieldaig. Because the pine and the birch, which is being used for regeneration, they form ectomycorrhizas. And just to give you a refresher of what these guys are, on the top left there, those are ectomycorrhizal tips of pine colonized by Amanita muscaria, very typically shaped, and the fungus completely encloses the root and then grows out from the root tip into the soil and take up nutrients. And you can see that on the right-hand side there. That's a little pine seedling colonized by a Suelus species, and the huge amounts of mycelium go out and take up nutrients, which are transferred. Some of them are transferred back to the seedling. And that's how these seedlings survive and grow. Okay? And the big trees are absolutely dependent upon these mycorrhizal fungi. So that's the required fungi for birch and pine. And many of the fungi involved are familiar to you, particularly during the autumn periods. You see the fruit bodies, and many of these are in fact ectomycorrhizal fungi. But at Ben Shieldaig, as I said, about 90% is actually heather moorland. And heather, like most plants in the Ericaceae, form ericoid mycorrhizas. The roots of these ericaceous plants are very odd. They're very, very fine. And they form what are called hair roots. And the individual cells of the roots are colonized. You can see this on the left-hand side there. The pink color is the fungus colonizing the roots, and you can see the individual hyphae going out into the soil and acting as absorbing agents. Okay, but these plants, the ericaceous plants, really have become fantastically evolved to maintain the conditions which effectively exclude other plants. They have very difficult recalcitrant liquor. It's full of phenolics and tannins, and it's only really the fungi associated with the ericoid plants that can use this liquor, and they effectively exclude other plants. Okay, so if we're wanting to plant ectomycorrhizal trees in here, we have a real problem. Similarly, a lot of the areas on the Ben Shieldaig are Millennia grasslands or rough grassland, And these guys form arbuscular mycorrhizal associations. Okay, so if you look at the roots of the grass or any other form in these systems, you will find vesicles, like the shown on the right here, there, or the arbuscules, which characterize these associations. And again, pine and birch cannot really form associations with arbuscular mycorrhizal fungi. Okay, so they're not going to get the support that they require if we plant them into these grasslands. So what we wanted to do was really to look at the site and to see what mycorrhizal associations were available, where could we potentially gain from planting birch and pine into appropriate vegetation. And the conclusion from the vegetation map was it's a major constraint because it's very limited availability of ectomycorrhizal fungi across this site. Okay, so that's quite an issue here. So what we decided to do was then do a detailed, very detailed field mapping of any potential sources of suitable ectomycorrhizal fungi across the site. And what we did was during October 2020, we, myself and a colleague here, Debbie Fielding, we spent 4 days walking across the site, and you can see the map on the left, and the thin red lines are our tracks, our surveys. And over the 4 days, we walked about 70 kilometers, and it's pretty rough ground, I have to say. We did about 8,000 meters of change in elevation, and we recorded about 450 observations on ectomycorrhizal plant species that may help establishment of any seedlings that we put in here. Okay? So we're trying to gain information on the ectomycorrhizal potential of plants across this site. Just to show you what we were checking out, we obviously recorded any birch, any pine seedlings that we came across, and also we recorded Arctostaphylos, bearberry. Now, some of you may know that Arctostaphylos is a fantastic ectomycorrhizal host, and it really supports a huge diversity of ectomycorrhizal fungi. So wherever we found that on the site, that was a real bonus. Unfortunately, it's very scarce on the site. We only found 3 or 4 patches, but they're certainly very important reservoirs of ectomycorrhizal fungi. Okay, so those were the first 3. But what we were really surprised to find was there was a lot of Salix plants across the site, both Salix aurita and Salix repens, and a hybrid, which I haven't shown here. And this is really, really important because Salix is very unusual in that it can form both arbuscular mycorrhizal associations and ectomycorrhizal associations. Okay, and we found hundreds of plants of Salix across the site. It was mentioned in the initial vegetation survey, but we did not expect to find hundreds of plants. So Salix repens, tiny little thing, only a few centimeters high, but very important because the Salix can spread right across the site wherever there is AM inoculum. Okay, wherever there are plants which support arbuscular mycorrhizal fungi, then the Salix can become established. And that's really important because over time, once it becomes established, that any ectomycorrhizal fungi arriving as spores can then colonize the Salix, and the Salix can then act as reservoirs of inoculum for ectomycorrhizal fungi of the birch and pine. Okay, so the Salix can spread right across the site because there is AM inoculum across the site, and then over time, as the ectomycorrhizal fungi arrive by spores, they can become established on the Salix, and then they can then colonize any birch or pine which lands in the vicinity. And that's super important. And what we found is that there's really strong evidence that the Salix forms groups, that's both within species and between species, and that it supports the establishment of birch and pine seedlings. We already have shown this on the site, because of the 32 birch seedlings that we found during the survey, the first initial survey, 31 out of the 32 seedlings that we found were associated with Salix. Okay? They're really only becoming established where the salix is already growing and where it supports ectomycorrhizal fungi. And that's really very important. So it's the networks, the ectomycorrhizal networks, which are extending out from the salix into the soil, which is supporting the establishment and then the growth of any birch and pine seeds which land, germinate, and become established. And this, we've looked at all sorts of things now, but the area of influence out from the salix is over 1 meter. Okay, it's just short of 1.5 meters. So, and when you have a big group of salix, you might have 20 or 25 or 30 plants, that actually gives you a big area that you can then use for planting. So that's really where we're at the moment. The next steps, obviously, is to take things a bit further. And we need to test the ideas, because it's all hypothesis at the moment. We want to determine the fungi that are actually on the salix roots. It's our hypothesis that they're supporting ectomycorrhizal fungi, but we don't actually know that yet. So we need to test that. We want to look at how fungal development is in relation to group size, if we can age the salix, because age will have a big impact on the amount of time that has passed in order for spores to arrive. Similarly, the distance from the existing woodland will be a big factor in the community development. So these things we're going to start looking at this year, and we'll use molecular tools to go below ground and to look at the fungi directly on the salix roots. In conjunction with this, we're working with the Woodland Trust to develop planting strategies which maximize the potential for these existing reservoir islands to act as inoculum to support seedlings. So we're working both in terms of using seeds, but spreading seeds in association with the groups, but also planting seedlings in amongst the groups as well. So that's what the next steps are. So hopefully, in a few years' time, the bare land will start to look something like this, with a mixture of pine and birch growing up. And I just wanted to acknowledge the team that's been involved in this. Debbie there, second from the left, she's been involved right from the start. And then last summer, there was 3 MSc students from University of Aberdeen, Paige Brown, Kerry Milligan, and Leon Marquez, who did their master's projects on the site, and they have gathered a lot more information regarding the positioning of, and the group size of the willows and the associated pine and birch. So that's where we're at, and thank you very much for your attention.
Thank you so much for your presentation.
Okay.
And we have a few questions from the audience as well. Åse asked whether it's possible to replant salix from one area to another to work as an inoculum in a way.
Yes, very much so. We're looking at all sorts of possibilities. I'm hugely in favor of collecting seeds from the local woodland and just wandering across the site and doing sort of broadcast sowing to look at in some areas and not others, and then measure the success of that. So there's all sorts of low input things we could do, which is important for a charity, obviously. But they're also looking at raising, they're going to set up a nursery. And one of the things that we could do is certainly grow up the salix. We're doing that in another project, in fact, but also with the Arctostaphylos, because it's a fantastic host for ectomycorrhizal fungi. So yes, we can certainly use the salix transplants.
I feel it's really like a bit of a chicken versus the egg discussion, because you need the fungi for the trees to establish, but then you need the trees for the fungi to thrive there.
Yep. Yeah, that's why salix is so important, because there's arbuscular fungi across the site. The salix can spread right across the site, and then it can sit there, growing quite happily. And then when ectomycorrhizal spores arrive, they can colonize. I've seen this, it's a similar phenomenon which I've seen in the tropics, but I've never seen this in temperate systems, I have to say.
Have you considered introducing ectomycorrhizal fungi in the nursery, or nurse these plants in an environment where there already are a lot of these and then move them out.
Yes, for sure. Ideally, there's an awful lot of work going on at this at the moment, and ideally, yes, you want the plants pre-colonized. And at the moment, nursery practices almost exclude the development of mycorrhizal fungi because the seedlings that are produced are stuffed full of nitrogen and nutrients, and when the plant's in that state, it rejects most mycorrhizal fungi. So I'm actually working with another charity called Trees for Life to look at nursery practices and how they can be adapted to promote mycorrhizal colonization in the nursery.
Yeah, all right. Håvard is also providing us with some information that there are some fungi that are able to make both ericoid mycorrhiza and ectomycorrhiza, and if these could be of importance for this reestablishment.
Yeah, for sure. There is quite a famous one that can form both. We've yet to find it, I have to say, in seedlings colonizing heathlands where it should be. I find it regularly on mature ectomycorrhizal systems, but I've also rarely found it actually in heathlands on the ericoid roots. So yes, there is a famous fungus that can do it, but it does not seem to be doing what we think it should do.
Right.
Yeah.
Margaret has a question. Is it an option to clear some of the headland in order to prevent this production of phenols and things that are there to prevent the colonization from other competing plants?
Yeah. One of the heather has been burnt in the UK for at least a couple of centuries, probably, and it grows back superbly well, and it actually maintains the dominance of the heather. You can scarify it, you can clip it, you can mow it, but it's amazingly resilient. The forestry used to spend a lot of money spraying around seedlings that were planted into heather moorlands because particularly citrus spruce is very prone to what's called heather check, and it will sit there doing nothing. They'll just sit there not growing for maybe 15, 20 years. And there's been a lot of work looking at that in relation to mycorrhizal effect on that. But there's certainly a big issue with stunting or check. It's very expensive to remove it.
Yeah, I understand. Is water access any issue in terms of reforestation? Because I imagine that on the top of this hill or mountain, if you want, is there enough water to support the birch and pine?
This is the West Coast. It's exactly the opposite. There's more than enough water. But most of the plants, because of the exposure here, 95% of the trees are below 200 meters. So it's quite far north. It's on the West Coast and it's quite exposed. The wind is a huge factor here. Up on the top, there's actually dwarf juniper, which is an interesting little thing, and it's right on the exposed top.
Yeah, all right.
Yeah.
Yeah, that was all the questions we had from the audience. Okay. Thank you so much for coming here and holding this presentation for us.
My pleasure.
All right, thank you.